Welcome to the class 2 video of bioinformatics. The most distinguishing characteristic of living things, their ability to store, utilize and pass on information. Bioinformatics strives to determine where what information is biologically important and to decipher how it is used to precisely control the chemical environment within living organisms. Since that information is stored at a molecular level, the relatively small number of tools available to molecular biologists provides our most direct insights into that information content. In this class, we discuss a brief introduction or review of the format in which genetic information is maintained and used by living organisms, as well as the experimental techniques that are routinely used to study in molecular biology laboratories. Now let us understand the genetic materials. DNA, deoxyribonucleic acid, is the genetic material. This is profoundly powerful statement to molecular biology. To a large extent, it represents the answer to questions that have been pondered by philosophers and scientists for thousands of years. What is the basis of inheritance? And what allows living things to be different from non-living things? Quite simply, it is the information stored in DNA that allows the organization of inanimate molecules into functioning living cells and organisms that are able to regulate their internal chemical composition, growth, and reproduction. As a direct result, it is also what allows us to inherit our mother's curly hair, or our father's skin color, or even our uncle's too bald head. The various units that govern those characteristics at the genetic level, be it chemical composition, or bald head, or nose size, long nose, etc., are called genes. Now let us understand the nucleotides in DNA. Genes themselves contain their information as a specific sequence of nucleotides that are found in DNA molecules. Only 4 different bases are used in DNA molecules: guanine, adenine, thymine, and cytosine. G, A, T, and C are the short form of this. Each base is attached to a phosphate group and a deoxyribose sugar to form a nucleotide. The only thing that make one nucleotide different from another is which nitrogenous base it contains, as shown in this figure. Differences between each of the 4 nitrogenous bases is fairly obvious even in representations of their structures such as those shown in this figure, and the enzymatic machinery of living cells routinely and reliably distinguishes between them. And very much like binary uses strings of zeros and ones, and the English alphabet uses combinations of 26 different letters to convey information. All of the information within each gene comes simply from the order in which these 4 nucleotides are found along lengthy DNA molecules. complicated genes can be many thousands of nucleotides long, and all of an organism's genetic instructions, its genome, can be maintained in millions or even billions of nucleotides. Now we are going to see the orientation of these nucleotides. Strings of nucleotides can be attached to each other to make long polynucleotide chains, or when considered on a very large scale, chromosomes. The attachment between any two nucleotides is always made by way of phosphodiester bond that connects the phosphate group of one nucleotide to the deoxyribose sugar of another, as shown in this figure. Ester bonds are those that involve links made by oxygen atom. That is, phosphodiester bonds have a total of two ester bonds, one on each side of a phosphorus atom. All living things make these phosphodiester bonds in precisely the same way. It is figured that each of the carbon atoms in a deoxyribose sugar has been assigned a specific numeric designation, that is 1 dash through 5 dash, by organic chemists. The phosphate groups of any single unattached nucleotide are always found on its 5 dash carbon atom. Those phosphate groups are used to bridge the gap between the 5 dash carbon of an incoming deoxyribose sugar and the 3 dash carbon of a deoxyribose sugar at the end of a pre-existing polynucleotide chain. As a result, one end of a string of nucleotides always has a 5' carbon that is not attached to other nucleotide, and the other end of the molecule always has been unattached 3' carbon. The difference between the 5' and 3' ends of a polynucleotide chain may seem subtle. However, the orientation it confers to DNA molecules is every bit as important to cells as our knowing that in written English we read from left to right and from top to bottom to understand the information content.